Hello everyone, welcome to another video where we will be covering biology paper 2B from November 2023. So let's get started. Question number 1 states, read the passage below, use the information in the passage and your own knowledge to answer the questions that follow. Alright, so let's first go to the questions and then we'll come back to the passage. A part, explain why more cars would result in an increase in atmospheric carbon dioxide. Alright, so as you know that cars use fuel or petrol or diesel, so you can mention that burning of fuel, burning or combustion of more fuel, or you can mention petrol or diesel, results in, or simply this is the answer. Because cars, they use more fuel, petrol or diesel, any one word can be used, and they burn it. So more burning or more combustion of fuel or petrol or diesel results in an increase in atmospheric carbon dioxide. B part, explain how increased carbon dioxide can cause climate change. Alright, so as you know that carbon dioxide is a greenhouse gas which causes greenhouse gas effect. And what happens is that it traps infrared radiation and hence causes global warming. So these are three valid points that we need to mention. Carbon dioxide is a greenhouse gas which traps infrared radiation. IR is a short form of infrared radiation. Resulting in global warming. C part, explain why increasing carbon dioxide concentration can increase photosynthesis in plants. Alright, so as you know that carbon dioxide is a raw material which is needed for photosynthesis. So increasing it can increase photosynthesis, whereas it can also become a limiting factor which can decrease photosynthesis as well. So you can mention that carbon dioxide is a raw material needed for photosynthesis and carbon dioxide can also be a limiting factor. So next part, explain why carbon sink depends upon respiration and deforestation as well as photosynthesis. So let's go back to the passage, lines 13 to 15. So lines 13 states, a carbon sink is an ecosystem such as a forest that absorbs more carbon dioxide than it releases. The size of carbon sink depends on the rate of photosynthesis, but also on the levels of deforestation and respiration. Alright, so we now know that carbon sink absorbs carbon dioxide, more carbon dioxide as compared to releasing it. So what happens is that since carbon sink absorbs more carbon dioxide than it releases, so respiration releases carbon dioxide, we know this, and deforestation releases carbon dioxide by combustion or by decomposition. So that's why it depends upon respiration and deforestation as well as photosynthesis. So you can mention carbon sink absorbs more carbon dioxide than it releases. Respiration releases carbon dioxide, whereas deforestation releases carbon dioxide by decomposition, or you can even say decay or combustion. Any one word can be used. E part, light affects gas exchange in leaves. Describe an experiment that shows how the net gas exchange from a leaf varies with light intensity. Alright, so we know that we can investigate this using hydrogen carbonate indicator. So what you need to do, you need to simply place leaves in test tubes with hydrogen carbonate indicator. Then you will place one leaf in light and one in dark, and then finally after some time you'll observe the color change and you'll see that indicator turns dark red or purple. And then also you need to mention the variables that you need to take: same size of leaves, same age of leaves, temperature, and all those stuff. So let's mention the points. First point will be place leaves in test tubes with hydrogen carbonate indicator. Second point would be place leaf in light and leaf in dark. And then finally third point will be the color change in light indicator would turn purple or dark red, whereas in dark it would turn yellow. And the last part, in that you can state the variables that you need to keep constant: abiotic and biotic. So biotic could be same size of leaf, species of leaf, and same age of leaf. And for abiotic you can mention same temperature, same volume of indicator, concentration of indicator, etc. So these are these are 4 valid points. Basically you can mention 5. You can take them as 5 because we have mentioned biotic as well as abiotic variables. So you need to mention any 3 of these in order to get the full marks. Next part, the photograph shows stomata on a small square of the lower surface of a leaf. The total area of the lower surface of the leaf is 150 centimeters square. Using the photograph and the total lower surface area of the leaf, estimate the number of stomata on the lower surface of the leaf. Alright, so as you can see here that in the diagram it's given that it's 0.4 mm. So what you need to do is that you'll multiply you'll multiply 0.04 centimeter with 0.04 centimeter to get the area for 2 stomata in this area of 0.04. So for 2 stomata, or you can even say 2 into 0.04 or 0.04 into 0.04, which will be 0.0016 centimeter square. Then you will find the density which will be 2 divided by 0.0016. Sorry, here it's 0.0016, not 116. That will be the density, and to calculate the total number, you will simply multiply 150 by the density. The whatever answer you'll get after dividing 0.0016 by 2. Whatever the answer that you'll get by dividing, you'll multiply that with 150, and the final answer will be 187,500 stomata. The final answer is 187,500. Next part states, stomata also have a role in water transport in the plant. Explain the role of stomata in water transport. Alright, so it is involved in transpiration, or you can even state water loss or diffusion or evaporation. You can use any one word out of these. And the second mark is for mentioning it draws water up from the roots into the leaf, and basically it creates a water potential gradient. With that, we are done with question number 1. Let's move to question number 2. Catalase is an enzyme found in many cells. This enzyme controls the breakdown of hydrogen peroxide into water and oxygen. A teacher uses this method to investigate the effect of pH on catalase. Step number 1, cut a cylinder of potato tissue into 6 equal sized discs. Step 2, measure 10 cm cube of hydrogen peroxide solution and place into a boiling tube. Step 3, add a pH buffer solution to the tube to keep the pH at 7. Step 4, add the 6 potato discs to the boiling tube. Step 5, collect the gas given off by the reaction in an inverted 20 cm cube measuring cylinder. And lastly, step 6, measure the total volume of gas collected after 5 minutes. Then it says repeat steps 1 to 6 using 4 different pH buffer, pH 4, 5, 6 and 8. And the diagram shows the teacher's apparatus. Part A, state what is meant by the term enzyme. Alright, so the term enzyme is, it is a biological catalyst, or you can even mention catalyst, that speeds up the rate of reaction without being used up themselves. So the important point over here is without being used up themselves. So you can mention biological catalyst. Biological catalyst that speeds up the rate of reaction without being used up in the reaction. Next part, explain why potato cylinder was cut into 6 discs rather than left as one cylinder. Alright, so cutting increases surface area, so that leads to increased speed of reaction. So you can mention increased surface area, which increases the rate of reaction or the speed of reaction. Next part, give the dependent variable in this experiment. So the dependent variable is the one that is change itself. So that will be the volume of gas that is released in 5 minutes. Next part, the teacher controls the time for gas collection and also the volume of hydrogen peroxide used. State 2 other abiotic variables the teacher should control in this experiment. Alright, so the abiotic variables can be temperature, and it can also be concentration of peroxide, or it can also be volume of pH buffer. You can mention any one out of these two, concentration and out of volume. Next part, the table shows the teacher's results. Calculate the percentage change in the mean rate of reaction as the pH is changed from pH 4 to pH 7. Alright, so we know that percentage change is final minus initial divided by initial into 100. So final value is 2.4. At pH 7 we have the rate of reaction in centimeter cube per minute 2.4. So 2.4 minus initial. At pH 4 we have 0.8. So minus initial divided by initial, which would be 0.8 into 100. So 2.4 minus 0.8 is 1.6. So 1.6 divided by 0.8 into 100. The answer is 200%. Next part, the teacher calculated the mean rate of reaction by collecting the oxygen released for the first 5 minutes of the reaction. Explain why the mean rate of reaction changes beyond the first 5 minutes. Alright, so what happens is that the substrate or hydrogen peroxide that is used up in the reaction and its concentration decreases. So what happens is that fewer enzyme substrate complexes are there are fewer collisions, and so water dilutes hydrogen peroxide. So you can mention substrate or hydrogen peroxide is being used up in the reaction, so its concentration decreases. This leads to fewer enzyme substrate complexes, or you can even state fewer collisions. And finally, water formed dilutes hydrogen peroxide. With that, we are done with this part. You can mention any 2 points out of these 3 and you will get the full marks. Next part and last part for this question states, explain the effect of changing pH on an enzyme control reaction. Alright, so we know that what happens is that as pH increases, the activity of enzyme increases until optimum pH. After that, it denatures the enzyme. The active site, the shape of the active site is changed, so enzyme and substrate, they no longer fit, and hence enzyme and substrate are no longer complementary. So you can mention these 3 points. pH as pH increases, the activity of enzyme increases until optimum temperature. Now here the important point is until optimum temperature. You need to mention this. Beyond the optimum temperature, optimum pH. Here also it is optimum pH, not temperature. Optimum pH. Beyond the optimum pH, the shape of active site is changed and substrate and enzyme no longer fits. With that, we are done with this part in this question. Let's move to question number 3. The diagram shows a nephron from a human kidney with some structures labeled. Which structure is the Broman's capsule? Alright, so we know that this structure, the Broman's capsule, is S, which is option C. So option C, S. Next one, which structure is the loop of Henle? So that is Q, which is option B. Next part, which structure is affected by ADA? So that is the collecting duct, which is P, option A. Next part, blood plasma contains much glucose but urine normally does not. Explain what happens to glucose in the kidney. So as you know that it is selectively reabsorbed by the process of active transport in the proximal convoluted tubule. So you will mention these points, that first of all, glucose passes from glomerulus into the Broman's capsule, and then it is selectively reabsorbed in the proximal convoluted tubule by the process of active transport. Now here, absorbed in the proximal convoluted tubule and by the process of active transport, these are two separate points, like two separate marks for these. And then finally, yeah, these are pretty much it. So the first mark that you will get will be that it passes from the Broman's capsule, it passes from the glomerulus into the Broman's capsule. The second mark is that it is reabsorbed. Third mark is in the proximal convoluted tubule, and another point that you can mention is by the process of active transport, to ensure that even if one point that you have mentioned is wrong, you still get full marks. So in such cases, make sure to write whatever you know, so that there's a chance, even if there's a chance that you might have made some mistake, so that your extra point overlooks that mistake. Next part, describe how a sample of urine could be tested for glucose. So we simply need to use Benedict solution. Then we need to heat it, and finally look for color change, red or green or even orange color show a positive result. So you need to mention that you need to use Benedict solution, then you need to heat it, and finally look for color change. If it is red, green or orange, that shows that glucose is present. Next part and last part, as a person sweats, they may become dehydrated. Describe the changes in the person's urine if they become dehydrated. Alright, so what happens is, if a person becomes dehydrated, less urine, or you can say lower volume of urine, is there, and urine is darker in color because it is more concentrated. So these are two valid points. The first mark is for mentioning that lower volume of urine is present, and the second mark is for mentioning that it is more concentrated. With that, we're done with question number 3. Let's move to question number 4. Mutation can cause changes in the enzymes produced during protein synthesis. State what is meant by the term mutation. Alright, so mutation means rare random genetic changes to the DNA. Next part, explain the different effects that a single base mutation can have on transcription and translation and the production and activity of an enzyme. Alright, so here you need to mention everything, since it has 5 marks, you need to mention the transcription makes mRNA, then mRNA will have different codons, different order of bases. Then you need to mention about tRNA, the different tRNA binds, they bring different amino acids, different sequence of amino acids is formed, different proteins, and then active site no longer fits if the shape of the active site is changed, and all of that. So you need to mention these points. Let's state transcription makes mRNA, then mRNA will have different codons, or you can say different triplets or order of bases, then different tRNA binds, or you can say different amino acids, then different sequence of amino acids is formed, then different sequence of amino acids cause different proteins to be formed, different enzyme. What happens is different enzymes, so the active site of the enzyme is changed, so active site no longer fits substrate. And the last point that you can mention is that mutation may have no effect as same amino acid can be used. As we know that different codons form same amino acids too, so that's why mutation can also have no effect. That is also a possibility. Now these are around 7 points. You need to mention any 5 of these to get the full marks. Next part. Scientists investigate the rate of spontaneous mutations in genes. They record the number of mutations per cell during mitosis and during meiosis. They count the mutations in 3 different strains of yeast and repeat each time the mutation rates are counted per division per cell. Calculate the mean mutation rate for strain 5207 during meiosis. Alright, so for mean we know that we need to add the values and then we need to divide by total number of values. So for the values we have 2 values, and the values are 132.6 132.6 and the second value that we have is 43.2. We'll add these both and the answer we'll get is 175.8, and then we'll divide this by 2 since the total number of values are 2, and finally the answer that we'll get is 87.9, and this is the final answer, 87.9. Next part, suggest whether mutation is calculated per division per cell rather than just per cell. So for here you need to mention that because of the 2 divisions in meiosis. So 2 divisions in meiosis. Next part and last part for this question, comment on the differences in rates of mutation for mitosis and meiosis in the different yeast strains. Alright, so let's go back to the table and see the differences. So here you can see that meiosis has higher mutation in all strains, whereas mitosis has lower mutation. Then you can mention that highest difference is there between mitosis and meiosis in 5207, whereas smaller difference between both of them in 5209. And then you can mention that highest rate for mitosis is in 5160 and highest rate for meiosis is in 5207. and then you can mention that there are differences between repeats of each strain. So you can mention these points. Let's write them down. Okay, so the first one you can mention is meiosis has higher mutation in all strains, or you can mention the opposite too, that mitosis has lower mutation. Next one, highest difference between mitosis and meiosis in 5207. Then smallest difference between mitosis and meiosis in 5209. Then you can mention lowest mutation rates for both of them in 5209. Highest rate for mitosis in 5160, and highest rate for meiosis in 5207. And last point that you can mention is differences between repeats of each strain. Now for such questions you just need to look at the table and you just need to tell that what is happening. Just you need to mention the highest, lowest, overall what is happening, in between, and all such things. But be careful that in such questions I have written in points form, but you need to mention in paragraphs. So with that we are done with question number 4. Let's move to question number 5. The diagram shows a nitrogen cycle with some processes labeled. Explain why plants need nitrate ions. So we know that nitrates are needed to make amino acids, and amino acids are used to make proteins. So to make amino acids and then to make proteins for growth. Next one, name the processes V, X and Y. So you can see here in the diagram that V is nitrogen fixation, X is nitrification, and Y is denitrification. So V nitrogen fixation, or you can say nitrogen fixing. X is nitrification or nitrifying, and Y is denitrification. Next part, students measure the dissolved nitrate levels in a river that passes through farmland. They also measure the nitrogen compounds being used on the farmland either as fertilizer or as manure from livestock. The graph shows how the nitrate levels in the river and the nitrogen compounds applied to the farmland changed from 1950 to 2005. Comment on the relationship between nitrate levels in the river and the nitrogen compounds applied to the farmland. Alright, so since they have stated here comment, so we also need to mention an explanation that whatever is happening in the table, whatever is happening in the graph, why is it happening. So first let's go back to the graph and see what's happening. So as you can see here that both of them, they have a similar pattern up to 1970. Both of them increased, and river nitrate, it is more stable than nitrogen on fields. Nitrogen on fields, it fluctuates. So you need to mention these 2 points for stating what is happening, and then you need to mention why it is happening for them to gain the next 2 marks. First let's mention this: both increase or both have a similar pattern up to 1970. Then river nitrate is more stable than nitrogen on fields, or you can mention that nitrogen on fields fluctuates. And then for explanation you need to mention that nitrate leaches into the river, or you can mention that it is washed into the river. And then you can mention that farmers, they add different amounts of nitrogen or fertilizers so different crops are grown. So with this you need to mention the first two points for telling what is happening, but regarding the explanations you just need to mention 2 points out of these 3 in order to get the full marks. Next one, describe the changes within the river that would have occurred between 1950 and 2000. Alright, so the changes that would have occurred would be more algal growth, or you can say eutrophication, or algae bloom, then more decomposers or more bacteria, then increased organic waste, or you can state less light penetration, then that leads to less oxygen in the river, and finally fewer fish species are there, or you can state death of organisms. Since oxygen is a raw material for respiration that all organisms need, so when there will be less oxygen, so less respiration, and hence organisms will die. So with that we are done with question number 5. Let's move to the next question. Question number 6, describe how stem cells are different from other cell types. Alright, so stem cells are first of all undifferentiated cells, undifferentiated or unspecialized cells that can become specialized, and they can become different cell types, and finally they can also continue to divide by mitosis to divide. Next one, stem cells from a donor can be used for stem cell therapy. Human blood cells are produced in bone marrow. Explain how using stem cells from bone marrow enables doctor to treat a number of different blood conditions. Alright, so you need to mention that they can make different blood cells like red blood cells or white blood cells or platelets, and then you can even say red blood cells, white blood cells or platelets. Then you can just mention the function of these, like red blood cells to treat anemia or to ensure oxygen transport around the body. White blood cells to improve immunity or to kill bacteria, and then platelets to treat blood clotting problems. So you need to mention the first point that they can make different blood cells, and then you need to mention any 2 points. Either state the function of red blood cells and white blood cells, or you can state the function of red blood cells and platelets, or white blood cells and platelets, or like 2 points from these 3 points. Next one, and the next part and the last part for this question in the paper, one type of stem cell transplant uses a patient's own stem cells. They can be used if the patient is producing enough healthy bone marrow cells. These cells can be collected, frozen, and stored for later use. Explain the advantage of using the patient's own stem cells rather than using stem cells from a donor. Alright, so what happens is that they will be genetically identical. They will, or you can say that they will have the same genes, so no rejection and no transfer of other disease or infections. So these are 3 points. You need to mention any 2 of them. With that, we're done with this part with the question number 6 and the paper. I hope this was beneficial for everyone, and let me know if you have any questions, any suggestions for future content, or like if you have anything to say in the comment section below. Thank you everyone for watching, and best of luck for your P2. Take care.